Hello students, you are watching Biology by Dr. N.S. Jain, and today we are going to study a topic known as the enzyme. First of all, you must know about that who gave the term enzyme. So the term enzyme was first of all given in the year 1878 by a scientist known as Willy Kuhn. Okay, and what is the literal meaning of this word enzyme? So the literal meaning of the word enzyme is in yeast. Okay, the literal meaning is in yeast, and you must know that these enzymes are biocatalyst. They are biocatalyst, and all the reactions, all the metabolic activities which are going in the cell cannot be performed without the enzyme. So enzymes are very important to perform all the metabolic activities. Now, there are few more discoveries given here. There was a scientist whose name was Buchner. So Buchner isolated a substance from yeast which was responsible for the fermentation, and this substance was known as the zymase. Okay, so Buchner was the scientist who first of all isolated the enzyme known as the zymase, and this substance or known as the zymase was responsible for the fermentation, and it was isolated from the yeast. Now the second is Sumner. Sumner isolated, or we can say that he isolated the urease enzyme in the crystalline form, means he prepared the crystals of the urease enzyme. And not only this, you must also know that all enzymes are proteins. Generally, I am not talking just now about the exceptions, otherwise maximum enzymes are proteins. So all enzymes are proteins, but all proteins are not enzymes. All enzymes are proteins, but all proteins are not enzymes, and the protein nature of the enzymes was first of all suggested by a scientist known as the Sumner. But here you see that I have written few exceptions, though we know very well that all enzymes are the proteins, but you can see that there are certain RNA enzymes also, which are only and only composed of RNA. They are not made up of the protein. See here, ribozymes and ribonuclease. P. Ribozymes and ribonuclease. These are called as the RNA enzymes. They are purely RNA enzymes. They are not having the protein. Okay, and they can catalyze, or we can say that they can catalyze the reaction even in absence of protein, means they are RNA enzymes. They are not having the protein. Okay, so all enzymes are proteins, but except the ribozymes and the ribonucleases, these are actually the enzymes which are made up of the RNA. Now, as we know very well that enzymes are acting as catalyst. Now, you study in chemistry also that catalysts are what substances which boost up the rate of the reaction, and they are not utilized in the reaction. So we can say that enzymes are organic catalysts. Which speed up various chemical reactions, right, without being used in it, or we can say that without being used themselves, the enzymes are acting as catalysts and they boost up the rate of the reaction. Okay, now the question arises that what are the substances on which the enzymes act? So you must know that the substances on which the enzymes act are called as what? The substrates. Okay, within few couple of minutes we'll be talking about the substrate also. First of all, you must know that the substance on which the enzyme act is called as what? The substrate. Now we will be talking also about the classification of the enzymes, as well as we will be talking about the nomenclature also. We will be talking about the nomenclature as well as the classification. If we see the classification of the enzymes, then the enzymes are actually classified in 6 major classes, and these 6 major classes are oxidoreductases, transferases, hydrolases, lysases, isomerases, and ligases. Okay, now these are all what? These are the classes. These are the 6 classes of the enzymes, and in these classes there are various subclasses also. So I have written few. I have not written all, but I have written few classes, few subclasses also of these six that is classes. See here. So this is the class oxidoreductases, and I have written few subclasses also. Dehydrogenases, oxidases, and reductase are the subclasses of the class oxidoreductase. Likewise, transferases, which is a class of enzyme, these are the subclasses of it. Transaldolase and transketolase. Hydrolases is another class, and it is having subclasses ribonuclease, phosphatase, etc. Lyases, aldolases are the subclasses. Isomerases are the class of the enzyme. Epimerase and isomerases are the subclasses. Ligases, the subclass is the synthetases. So these are the classes of the enzyme, and these are the some subclasses of the enzymes. Okay, so you must know that what is the nomenclature also. So if we put a suffix ASE, maximum enzymes, I am not talking about all, but maximum 99% chances you will see that there is a suffix ASE added at the end of the enzyme. You can see lactase, maltase, sucrase, okay, amylase, okay, all these enzymes are having ASE. So what is the nomenclature of the enzymes? That a suffix ASE is added. ASE is added at the end of the enzymes. Okay, now the question arises that some more about the enzymes, some more about the enzyme. So you must know that an enzyme is not always purely made up of only the protein. No doubt at all, there are some enzymes which are purely made up of the protein, but maximum enzymes are the conjugated proteins. Maximum enzymes are the examples of what? Conjugated proteins. So first of all, you must know that enzyme, if we talk about a holoenzyme, then what is a holoenzyme? So holoenzyme is what? It is made up of apoenzyme. It is made up of apoenzyme plus cofactor. Now what is a holoenzyme? Holoenzyme means what? Complete enzyme. The complete enzyme is called as what? A holoenzyme. And a complete enzyme, or also known as we can say it is a conjugated protein. A complete enzyme is called as a conjugated protein. Means what? Apoenzyme is the protein part of the enzyme. Apoenzyme is the protein part of the enzyme, and the cofactor is the non-protein part of the enzyme. Non-protein part of enzyme. Means what I want to say, that maximum enzymes are having protein part also, as well as they are having non-protein part also. Maximum enzymes are having protein part also, as well as the non-protein part. But I am giving exception. I am giving you the exception here. There are few enzymes. You can watch here that pepsin, amylase and urease. Pepsin, amylase and urease. These are the enzymes which are purely made up of the proteins. Means I want to say pepsin, amylase and urease enzymes are not having the non-protein part. They are not having the non-protein part. They are only purely the protein enzymes. But maximum enzymes are having protein part also, as well as the non-protein part also. Right? Now the question arises that these cofactors are of two types, organic and inorganic. These may be organic or inorganic. These cofactors may be organic or inorganic. In the case of the inorganic cofactors, ions are included. You can see that many times Zn++, Mg++. There are various inorganic cofactors in which ions are included. Okay, so ions are participating here. While in the case of the organic cofactors are of two types. Okay, one is known as the prosthetic group. And another is known as what? The coenzymes. Another are known as coenzymes. One is known as the prosthetic group, another is known as what? The coenzyme. Now, what is the difference between prosthetic group and the coenzyme? So you must remember that prosthetic groups are firmly attached to apoenzyme. They are firmly attached to apoenzyme, which is the protein part of the enzyme. Prosthetic group is a group which is firmly, tightly attached to the protein part of the enzyme, which is also known as the apoenzyme. And what about the coenzyme? So they are loosely or transiently means for a while, not permanently, for a while. They are loosely or transiently bound to apoenzyme. They are transiently or loosely bound to the apoenzyme. Okay, so there are many coenzymes such as NAD, NADP. Okay, these all are what? Coenzymes. These all are coenzymes, and prosthetic groups are firmly attached to the apoenzyme. So a holoenzyme is made up of a protein part as well as non-protein part. Non-protein part of the enzyme is cofactor. Cofactors may be organic or inorganic. In inorganic, ions are used, right? And organic cofactors are of two types: prosthetic group and coenzyme. The group which is firmly attached, tightly attached to the apoenzyme is called as the prosthetic group, and the group which is loosely or transiently bound to the apoenzyme are actually called as what? The coenzymes. Now we are coming to the next one, that enzymes are classified by one more method, by one more way, and that is that where they are present, based on where they are present. Either they are present inside the cell or outside the cell. On this behalf, we can say that enzymes are classified into two categories. Enzymes are classified into two categories. The enzymes are known as, some enzymes are called as endoenzymes. Maximum enzymes are endoenzymes, while some enzymes are exoenzymes. Right? So endoenzymes are what? Are the enzymes secreted within cells. Say for chloroplast. Chloroplast have many enzymes within it, and it helps in photosynthesis. Likewise, mitochondria have many enzymes in it. And it helps in respiration. So endo, as the name is indicating, endoenzymes are the enzymes which are secreted within the cells, while exoenzymes are secreted outside cell, right? They are secreted outside cell in medium by cell membrane. Are secreted outside cell in medium by the cell membrane. These are actually found in, exoenzymes are basically secreted by certain bacteria and by certain fungi. Okay, by certain bacteria and fungi. Here the enzymes are secreted outside the medium, right? Outside the cell, outside. Okay, they are secreted outside the cell in some medium. So these are the two categories, endoenzymes and exoenzymes. Now we will be discussing about the properties. We will be discussing about the properties of enzymes. That what are the properties of enzymes? So here is the list of properties. Number one, enzymes are acting as the biocatalyst. They are acting as what? Catalyst. As we know very well that they boost up the rate of reaction without being used up in it. Number 2, maximum, I am not talking about the exceptions, they are of protein nature. They are of protein nature. Enzymes are very much specific. Enzymes are very much specific. They cannot be replaced by some other. They cannot be replaced by some other. Say for if the salivary amylase is acting in the buccal cavity, then you cannot replace another enzyme for that one. Okay. So they are very much specific. They are colloidal in nature. They are colloidal in nature and provide they are colloidal in nature and provide larger surface area for the reactions to go on. Right. 5th one, biocatalyst, they are of protein nature, they are specific, they are colloidal in nature, they have high molecular weight. Their molecular weight is very much high. Okay. Next, they are actually, you know very well that they are protein nature, so they are amphoteric. Enzymes are amphoteric. That is they can act on acidic as well as alkaline. They can act on acidic as well as alkaline substrates. The next property is enzyme optima. Enzyme optima means enzymes are very much sensitive for pH and temperature. Next we'll be talking about enzyme inhibitors. Enzyme inhibitors, there are certain substances, certain substances like insecticides, drugs, etc., which can inhibit enzyme action, and the next one is enzymes can act in both direction. Means they can catalyze the reaction in A to B or B to A, which is also called as reversibility of reaction. Means enzyme can act in both direction. Not only they can catalyze the reaction from A to B, they can catalyze the reaction from B to A. So these all are the major properties. of the enzymes. Okay. Now after talking about the properties, we will be talking about the mechanism of enzyme action. That how do the enzymes act, okay, or how do the enzymes work. So we are talking about mechanism of enzyme action. Whenever we talk about the mechanism, we have to study two things. First of all, enzyme substrate complex. We have to study here that how an enzyme substrate complex is formed. And second is that enzymes are having the role of lowering activation energy. Enzymes are having the capacity to lower the activation energy. Just of now we will be talking about this. Now first of all, enzyme substrate complex. You must know before starting the enzyme substrate complex, you must know that enzymes have certain pockets. Enzymes have certain pockets, right, which has what? Amino acids, which have amino acids, right? Amino acids arranged in them, which have amino acids arranged in them. Enzymes have certain pockets which have amino acids arranged in them, and these pockets are called active sites, where substrate is attached, where substrate is attached. Now, when we are talking about the mechanism of enzyme action, you must know that how enzyme substrate complex is formed. Say for I am making an enzyme. Say for this is an enzyme. And what are these? These are the active sites or the pockets where the substrate will be attached. These are the active sites, and this is what? This is a substrate. This is a substrate. So how the substrate is formed? Like this enzyme substrate complex is formed. Now this is enzyme, and this is what? Substrate. And these are the active sites. In these active sites, the substrate will fix like this type. The substrate will fix and it will form, this is enzyme, this is substrate, it will form enzyme substrate complex. This will form what? Enzyme substrate complex, and at last you will get the product. Plus enzymes are not changed, right? Enzymes are not changed, and they will be regained back. Like this enzyme substrate complex is formed. Like this, what is formed? Enzyme substrate complex is formed. Now, to explain the phenomenon of the enzyme substrate complex, you must study, right, two models which will betterly explain the formation of enzyme substrate complex. So to study formation of enzyme substrate complex, two models are available. One is known as lock and key model. And second one is known as induced fit model. First of all, we are going to study the lock and key model. First one, lock and key model. It was given by Fisher. What does this theory says? That just like a particular key fixes in a particular lock, exactly in the same manner, this theory says that the active sites of the enzyme and the substrates are having the same type. Okay, they are having the same sites. So the substrate will go and fix at the active sites of the enzyme, and like this we can see that a lock and key model will be seen. Say for if I am making an enzyme here, see here I have made an enzyme here. This is an enzyme, and these are the active sites. Okay, now this is what? A substrate. You can see here that just treat this enzyme as a lock and just treat this substrate as a key. Just like a particular key fixes in a particular lock exactly. Here the substrate and the enzymes are having the same sites. So this substrate will go and fix at this site just like in a lock and key fashion. So what will happen in lock and when they will be attaching together in lock and key fashion, what will happen? That enzyme substrate complex is formed. So this is E and this is S. This is known as ES complex, enzyme substrate complex. At last you will get what? The product. You will get the product plus enzymes are regained back. Enzymes are regained back. So this is known as what? Lock and key model, which was given by Fisher. But we are having more defined model. We are having more defined model rather than this lock and key model. Nowadays, lock and key model is not accepted because we are having a more favorable that is known as a model for enzymatic action, and that is induced fit model. That is known as what? Induced fit model, and it was given by Koshland. It was given by what? Koshland. What does this model says? What does this model says? This model says that in the beginning, this model says that in the beginning, the sites which are present on the enzyme are not similar, are not at all similar to the site of the substrate. You can see here, you cannot see or you cannot assume that just like a key particular fixes in a lock, here you can see that the sites of the substrates and the enzyme are totally different. But what does the induced fit model says? Induced fit model says that when substrate come near enzyme, so the site, or we can say that the membrane of enzyme undergo certain conformational changes, and become similar to substrate, so that the substrate will fix in the active site of the enzymes. See here, initially you can see that the site present on the enzyme and the site present on the substrate are totally different. But as soon as the substrate will come near the enzyme will get induced, and the surface of the enzyme, that is the membrane present on the enzyme, will undergo certain configurational changes and it will become similar to the substrate. So ultimately what will happen? The enzyme site will become similar to that of what? Substrate, and ultimately you can see that what will be formed? A enzyme substrate complex form. So this is enzyme substrate complex. This is enzyme substrate complex formed, and ultimately you will get the product and enzyme is formed. So which is the best model to explain the mechanism? So lock and key model is an older model. Induced fit model is a very good model to explain the mechanism of the enzyme action, or if we want to study that how enzyme substrate complex is formed. So this is rather a good model in comparison to the lock and the key model. Now in the second part we have to study the lowering of activation energy. Lowering of activation energy, very important part. Lowering of activation energy. Lowering of activation energy. First of all, you must know that each and every reaction, whether it is an exergonic process also, it liberates energy. Say for if we talk about a exergonic process, say for if I talk about combustion of wood, if I talk about the burning of the wood, then when the wood burns, okay, then initially what was required for the burning of the wood? For the burning of the wood, a small ignition was required, right? So what I want to say that each and every reaction requires a small amount of energy to be ignited or to be started. Okay, so molecules molecules before reacting require a activation energy. Molecules before reacting require a activation energy. Means each and every molecule, if a molecule has to react, then it will be requiring a small amount of energy, and that energy is called as what? The activation energy. Molecules before reacting require a activation energy. But why we are studying activation energy in the case of the enzymes? What is the role in activation energy, right, of the enzymes? So always remember, enzymes lowers activation energy, which is required by molecules to react. Enzymes lowers activation energy. Remember, this question is asked many times, that enzymes, what is the role in the activation energy of the enzymes? So enzyme lowers activation energy, which is required by molecules to react. Say for, this can be represented by a graph also. Say for, there is a reaction like this type, right? This is a enzyme catalyzed reaction. This is a enzyme catalyzed reaction. This is an enzyme catalyzed reaction, where you can see that lower activation energy. You can see the peak. Lower activation energy. Okay. But when this is an enzyme catalyzed reaction, but when enzyme is not present in the reaction, you can see here that you can see the peak. Here. You can see the peak. This is an enzyme catalyzed reaction. An enzyme catalyzed reaction requires a lower activation energy, while this is what? This is reaction not catalyzed by enzyme. This is a reaction not catalyzed by enzyme. This is the reaction not catalyzed by the enzyme, and you can see that activation energy is higher activation energy. This is what? Higher activation energy. So you can see that an enzyme catalyzed reaction is having less requirement of the activation energy, means lower activation energy. But when a reaction is not catalyzed by the enzyme, then it requires what? A higher activation energy. So each and every process requires a small amount of energy to be ignited, to be started, and that energy is called as what? Activation energy. Similarly, molecules before reacting require a small amount of energy, which is called as activation energy. What is the role of the enzyme? That enzyme lowers the activation energy, which is required by the molecules to react. Okay, so these were the two concepts: formation of the enzyme substrate complex, right, in the mechanism of enzyme action, and second is the lowering of the activation energy. Okay, now we'll be talking about the inhibition also. We'll be talking about inhibition. What is inhibition? Inhibition means what? Stoppage, right? Certain substances inhibit. Certain substances inhibit action of enzymes. Certain substances inhibit action of enzymes. Now this inhibition is of two types. One is known as competitive inhibition, and another is known as what? Non-competitive inhibition. Now, what is competitive inhibition? Here there is a competition between substrate and inhibitor. To attach with active site of enzyme as the substrate and inhibitor are almost they are same. Now the question arises that who will attach, whether the substrate will attach or inhibitor will attach to the enzyme. So substance which has higher concentration will attach to enzyme, if inhibitor is having high concentration, then it will attach to enzyme. We are talking about what? Competitive inhibition. Now think of that this is an enzyme. Now see here that this is a substrate, and this is a inhibitor. I means inhibitor. So both are same. Now this is what? Substrate. Now exactly substrate and inhibitor are seeming to be same, and this is what substrate, this is what inhibitor. Now question arises that what happens, that when they are same, a competition is held between the substrate and the inhibitor, that who will attach with the enzyme. So what I want to say, that substance will attach to the enzyme whose concentration is high. Say for if its concentration is high, if concentration high, so what will happen? The substrate will not attach and inhibitor will attach, and instead of the formation of the enzyme substrate complex, a new complex will be formed, right, like this type. A new complex will be formed, and this is enzyme and this is what? Inhibitor. So this is what? Inhibitor. So instead of ES complex, enzyme substrate complex, EI complex will be formed, that is enzyme inhibitor complex will be formed. Now if we remember, if we increase substrate concentration, then inhibitor can be removed. Say for if we want to remove inhibitor from the enzyme, then you can increase the substrate concentration. If you increase the substrate concentration, this inhibitor will be removed. Like this type, competitive inhibition will occur, and when inhibitor will combine with the enzyme, then the product will not be formed, right? Product will not be formed because inhibitor has attached to the enzyme. But what happens in the non-competitive inhibition? So in non-competitive inhibition, there is no direct competition. Of a substrate with the active sites. Okay, no direct competition of substrate and inhibitor. Inhibitor will attach at some other site of enzyme. See here, say for this is a enzyme, okay, and this is a, say for inhibitor, say for this is the substrate, so the inhibitor will not attach at these active sites, inhibitor will not attach with this active sites, instead inhibitor will attach at some other site of the enzyme. This is what inhibitor. See here that inhibitor is attached here, instead of attaching at the active site, inhibitor has attached this. So this was about the inhibition. that there are certain substances which inhibit the action of enzymes. Inhibition is of two types, competitive and non-competitive. At last we will be talking about the factors affecting the enzyme action. Factors affecting enzyme action. Factors affecting enzyme action. The very first is, that is substrate concentration. Substrate concentration. Always remember, if we increase substrate concentration, then what will happen? If we increase substrate concentration, then enzyme action will be increased. Okay, if we increase what? Substrate concentration. What is thing? What we are studying? Factors affecting enzyme action. If we increase substrate concentration, so what will happen? A rate of reaction increased initially, but a time will come when enzyme become limited. Okay, so what first of all I want to say, that substrate concentration, if we increase the substrate concentration, a rate of reaction is increased initially, but a time will come when enzyme becomes limited. So if you are increasing substrate concentration, it's all right, but if you are not increasing the enzyme concentration, a time will come when enzyme will become limited. And what will happen, that after that, if you are increasing the substrate concentration also, then there will be no impact on the rate of reaction. Second one is, that is enzyme concentration. Second one is enzyme concentration. By increasing enzyme concentration, a rate of reaction increase initially, but a time comes when substrate become limited. Now the point to be noted here is that substrate become limited. Here in the above one, what you have seen, that enzyme has become limited, but here you can see that substrate becomes what? Limited. So if you increase the enzyme concentration, no doubt at all reaction rate will increase, but up to a limit, because a time will come when substrate will become limited. And what about the pH? Always remember that enzymes work at optimum pH. Generally they work between 5 to 9 pH, but it's not necessary that all are acting at this pH only. No doubt at all, they are very much sensitive about the pH, but there are certain enzymes which work at a particular pH only. Say for it's not necessary that they will be working between 5 to 9 only. Say for there are some enzymes, for example pepsin, which is found in the stomach, it is working at 1.5 pH, which is highly acidic. Okay, so it's not necessary that they will be working in between these only. Okay, so they are very much sensitive to the pH, and you must also know that they are very much sensitive to temperature also. They work at optimum temperature, and this optimum temperature is between 5 degrees to 40 degrees. Okay, below 5 degrees and above 40 degrees they are not working. In the case of the human beings also, if we talk about the human beings also, when the fever comes, what happens at the time of the high fever, our body stops working. Why? Because at high fever all the enzymes stops working and the body do not respond. Okay, in the plants also, right, certain enzymes are there, and that enzymes do not work above the 40 degrees temperature. So I want to say that temperature, there is an optimum temperature for the enzyme to act. Okay, and they are very much sensitive to this. So these are the factors: substrate concentration, enzyme concentration, pH and temperature are the major factors which are affecting the enzyme action. So dear student, this was the whole video based on the enzymes. Okay, keep watching my videos. Thanks a lot.